Well, I think it's fair to say that the seed grant that I got from the HSCI really changed the course of my whole career. I can't say it too much more simply than that. When I first started here as a Harvard Junior Fellow, about to become an assistant professor in the Department of Molecular and Cellular Biology, Doug Melton and I together had this big idea that we would try to use embryonic stem cells to try to really develop a new type of disease model that had never existed before. And that was to begin to use stem cells to make the actual cells that were getting sick in a disease and see whether or not they would begin to tell us a story about why those patients began to get sick. And initially, it was impossible for me to develop any sort of funding stream for this experiment. I had no history at all in studying neurological diseases, and this idea was completely, seemingly outrageous at the time. We were fortunate enough to receive that grant, Paolo DiGiorgio and I, my first graduate student, and Paolo immediately set off with that seed grant to really test whether or not he could make, at first, mouse and then human embryonic stem cell models of amyotrophic lateral sclerosis, or ALS in the United States, known as Lou Gehrig's disease. And what he showed in those first experiments that were funded by the seed grant was that indeed he could take advantage of stem cells to make very large numbers of spinal motor neurons, which are the neurons that die in ALS. And that, I think even more importantly and more surprisingly, in some of those neurons he could begin to see what are really thought of as being hallmark pathologies that are found in Lou Gehrig's patients. And even more than that, I suppose, of greatest significance was that he really learned something unexpected and surprising about the disease. And that was that despite everyone else's thinking that really motor neuron death in this disease was sort of like suicide of those neurons, that instead, or maybe in addition to that, there were other cells in the nervous system which were taking on an almost murderous type of quality. And that is that they were coming after and even killing the motor neurons in the context of this disease model. And that discovery has actually now been shown by many other groups to play out in the animal models of the disease in other contexts, and is now thought of as being a leading therapeutic target for the disease ALS. And really with subsequent funding that was made possible by that first seed grant, we've gone on to discover new chemical compounds which seem to have dramatic effects in those disease models. And one of the things that we're doing now, one of the things which is coming next, is that we're actually testing those different compounds in an animal model of ALS. And if they're successful there, that would be the first major step towards taking those compounds that we discovered using stem cells into the clinic and testing whether or not they could become new drugs for ALS. So I think there's really no doubt that that finding by Paolo and myself has had a substantial effect on the ALS field itself. It's also had a very, of course, substantial effect on the course of my laboratory and the types of experiments that we've been able to do. I don't think that there is much of any doubt that those experiments were a major contributing factor to being able to receive now large grants from the NIH, significant support from a number of disease-specific foundations, including the New York Stem Cell Foundation and Project ALS. But maybe I would say most excitingly for us, it's those experiments that were really the cornerstone of our application for this Early Career Investigator Award for the Howard Hughes Medical Institute. And receiving that award, as we did several years ago now, is really kind of the cornerstone funding for the laboratory that allows things to go forward.